Welcome everyone to Chem 114 GOBC. Okay, we're talking about lipids today, and lipids are a broad class of molecules. We've just went into esters and carboxylic acids, which are made. We make carboxylic acids, for example, from aldehydes when they're oxidized. Then we learned how we can make esters from carboxylic acids and alcohols coming together and the mechanisms, well, not the mechanisms, but the reactions that actually help produce those molecules. Although they're simple organic chemical reactions, our enzymes can do many of these things. Enzymes throughout many organisms, from bacteria to plants to us human beings. Enzymes do the chemistry that we often do in the lab. And the world of chemistry is growing to where enzymes can be used outside of a living organism to actually do chemistry. So these are all the neat things that the future holds for us. So let's get into lipids. Lipids are a broad class of molecules. They can do everything to make our membranes that give us that can sequester and organize cells and provide a divide from what's aqueous and non-aqueous to becoming a hormone that can actually create biological signals. And we have things like cholesterol that do many, many things that can even act as a damper in cell membrane fluidity and how easily a cell membrane can move. So cholesterol, although as you hear about many things about cholesterol, it's actually a wonderful molecule that us humans need. So let's go ahead and explore lipids. So we're exploring lipids, and um, we have heard before about what they call HDL and LDL, and there can be lipid tests, and patients can be examined for their HDL or LDL. HDL tends to be the problem. LDL tends to be the good form of cholesterol. HDL tends to be the bad form of cholesterol when it gets too high. We know high triglyceride content sometimes can lead to obesity and even coronary heart disease, but there's more factors than just the fact of lipids. Lipids are absolutely needed for our survival. Um, Two classes really of lipids. The fatty acids, which we're sort of already familiar with, they're the backbone of lipids, and we're going to learn that the true backbone of a lot of lipids is the glycerol molecule, where alcohols. Remember, I taught you all of you about carboxylic acids and alcohols coming together to form an ester. We didn't do that for no reason. We did that to explain to you that glycerol, which is a it has three alcohol groups on it, often will react with fatty acids and will be connected to the glycerol alcohol molecule, and there'll be esters formed with these long chain fatty acids that are in our membranes that make up our membranes. So lipids can be fatty acids. They can be the steroid nucleus. This is just a rough sketch of what most steroids that are based on cholesterol are going to look like. Um, so a steroid, if they're talking about a steroid, they're talking about something that often just is similar to cholesterol. Um, all of these lipids, all of them are usually soluble in organic solvents, unlike sugars that like water because they're so hydroxylated, they have so many alcohol groups. Notice there's not a whole lot of alcohol groups shown on the steroid nucleus. It's pretty much carbon and hydrogen. Here, this fatty acid chain is way longer than 6 carbons, and that's how we often classify fatty acids, by how many carbons they have. And for example, this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. It's not going to be soluble in water. It's way past 6 carbons, and it's going to be fairly insoluble in water. And the steroid nucleus, the same thing. It's going to be insoluble in water. Lipose means the word fat from the Greek word, and it's very important in cell membranes for fat soluble vitamins and steroid hormones. The difference between non-fat soluble vitamins and fat soluble vitamins are the body's retention. Fat soluble vitamins are often retained in the body for a much longer time than, say, vitamin C. Vitamin C is like a little bit like sugar, and it is easily dissolved into the aqueous milieu. Where when we talk about these steroids, when they get into fats, stored fats, they stay for a long time. That's why it's much safer to overdose on a vitamin that's water soluble. If a vitamin's water soluble, like vitamin C, you're pretty safe. If you take too much, it'll just be kicked out of your system. But if you take too much of like a vitamin E or a vitamin K, those are vitamin A, vitamin E, vitamin K, all are fat soluble vitamins, and they stick in the body much longer because they dissolve in the non-aqueous, non-polar fatty part of the body, and they get stored there for a long time, so they can be more toxic. Um, so moving on to these two classes of lipids, fatty acids and steroids. So lipids can be classified as waxes, fats, oils, and there's also phospholipids. We can phosphorylate this chain here, or we can phosphorylate the glycerol, which you'll see that later. Now remember, glycerol molecule has three hydroxides on it, three alcohol groups that can react with three different fatty acids. And it'll often react with two fatty acids and be phosphorylated at the third alcohol in general. Steroids, also liquids, do not contain fatty acids. They can't be easily hydrolyzed. Um, Fatty acids can be chewed up for energy when necessary. Um, these are this is what they call the steroid nucleus. Almost all steroids go through this in the human body, this kind of a backbone. So very unique structure, and does many different groups can be placed on this cholesterol steroid-like molecule, this steroid nucleus, and can do many different things, including signaling and activating genetics, and all sorts of stuff can come from steroids. So really cool stuff. And so getting into this more in depth, what I was talking about is, um, so now you can see it up close. This glycerol molecule, the backbone of these fatty acids and these lipids. These lipids make up our cell membrane. And so glycerol here, see how it has a hydroxyl here at each end? There's a hydroxyl, there's three hydroxyls. Now guess what? The chemistry you learned earlier is very important, because you remember we said a carboxylic acid and alcohol can react to form an ester. Well, these lipids that make up our cell membrane do exactly that. And this fatty acid, you could put one, two, or even three fatty acids on glycerol. And then that would be a molecule that separates the membrane from its aqueous environment, right? The cell membrane has to have some way to have integrity. Fatty acids can come from long chain alcohols, fatty acids. Triglycerols, that's what they're speaking of here, where glycerol has three fatty acids attached, one, two, three. Now, there's also the specialized glycerol phospholipids. They only have two fatty acids. Then they have a phosphate group, PO4. Remember we talked about polyatomics, um, polyatomic anions often. Well, phosphate was one of those, and it can be attached at one of the alcohols, and often it's at the end, and then it can be attached to an amino alcohol. So you can have two fatty acids, a phosphate, and an amino alcohol all attached to a glycerol molecule, adding for complexity. And so when we talk about an amino alcohol, we kind of see that right here. See, there's an amine group and a hydroxyl group, and then there's some methyl groups. So um, next up, we have sphingomyelins. These are in nerve tissue and such, and these also can use interesting molecules, as you see here. Instead of glycerol, it has this long, sophisticated sphingomyelin that's has an amine, a phosphate, a hydroxyl, a mid group. a double bond, quite sophisticated. I don't expect you to memorize this, but kind of be aware of all the groups that are in sphingomyelin. Then it can be attached to fatty acids, which is one of these, can go here, and then a phosphate and an amino alcohol, as we talked about with glycerophospholipids. So these are naturally occurring biomolecules. They're very soluble in organic solvents, but not in water because there's so much carbon and hydrogen relative to the oxygen that it's a fairly nonpolar molecule, even though there is a little bit of polarity here as I'm circling, even though there's a little bit of polarity here, it's pretty much a non-soluble, um, non-polar molecule in general. But now this comes in handy because if there is going to be interaction between the aqueous environment and the non-aqueous environment, it may come at areas like this where there is some polarity, right? Okay, so think about polarity. Remember the Linus-Pauling scale of electronegativity. So, how do we characterize these fatty acids and steroids, and which are both lipids? And so, if we see something that contains a fused four-member ring system, we should automatically say, it's going to be the steroid nucleus, which are a bunch of rings. Contains a long carbon chain, and it's a lipid. It's probably a fatty acid. Includes carbonyl groups. That's probably a fatty acid, although some of the steroids may contain one or two, depending on how they're modified, but rare. So the first answer is steroid, second is fatty acid and fatty acid. So that those are some of the features to think about with lipids and the two classes of fatty acids and steroids. How can you determine which is which? By functional groups, right? Functional groups. Carbonyl groups often are just on the fatty acid at the carboxylic acid, or the carbonyl that attaches to glycerol, right? When it forms an ester. Okay, so which of the following is not a characteristic of lipids? Well, may contain a carboxylic acid group. May contain a four-ring structure. It's soluble in water. It's mostly nonpolar. All of these are true except for one. It may contain a carboxylic acid group, sure. Carboxylic acid reacting with alcohol with a fatty acid. May contain a four-ring structure. Yes, the cholesterol steroid molecule or nucleus. Soluble in water, we should say no. They're pretty much not soluble in water. D, mostly nonpolar. Well, that's the definition of insolubility in water. So C must be the right answer. Coming up with the negative. Lipids are nonpolar and therefore they're not soluble. And I thank all of you. This has been GLBC Chem 114 Introduction to Lipids. We'll be back soon.